Good morning. I am Gothaiammal, working as an assistant professor in Department of Microbiology. Today we are going to learn about medical parasitology. Before we start, let's look at what we are going to learn today. First, define parasitology, then well understand why this topic is important, especially how parasites can cause the disease in humans and why we need to know about them. First, we know the definition of medical parasitology. Medical parasitology is the branch of science that deals with the study of parasites that cause the disease in humans, including their life cycle, mode of transmission, pathogenesis, clinical features, diagnosis, treatment and prevention. Next, we know about the scope of medical parasitology. First, identification and classification of medically important parasites. It involves detecting and categorizing parasites based on morphology, genetics and life cycle for accurate diagnosis and treatment. Second one, understanding the host-parasite relationship. Examine how parasites interact there with host, affecting immunity, survival and disease progression. Then studying the life cycle of parasites. It focuses on tracking the developmental stages of parasites to identify transmission route and vulnerable points for intervention. Next, diagnosis and treatment of parasitic disease. It entails laboratory testing and clinical methods to detect infections and administer effective antiparasitic therapies. Next, developing the strategies for prevention and control. It involves designing the public health programs, vector control, sanitation and vaccination plans to reduce the parasitic infections. Last one, public health awareness and surveillance. It promotes education, monitoring and reporting the system to prevent outbreaks and ensure timely response to parasitic threats. Next, we can see about the importance of medical parasitology. First one, global health impact. Parasitic diseases like malaria, amoebiasis, leishmaniasis affect millions globally. Many are endemic in developing countries due to poor sanitation and hygiene. Tourists and travelers are at risk of contracting parasitic infections, zoonotic transmission of some parasites. Next, we can see about the types of medically important parasites. There are three parasites important for medically, that is protozoa, helminths and arthropods. Protozoa is a single celled organisms, example Plasmodium, Endamoeba histolytica. Helminths, it is the multicellular worms, example Ascaris, Taenia, Schistosoma. Arthropods, it is the insect and arachnid that act as a parasites or vectors. Example of the arthropods, that is the mosquitoes, ticks and lice. Next, we can see about the host parasitic relationship. Parasite is nothing but an organism that lives on or in another organism, that is called as the host, and it gets food from or at the expenses of the host. Host is nothing but which organisms that provides nourishment and shelter to the parasites. Types of host. There are four different types of host, that is first one, definitive host, second one, intermediate host, third one, reservoir host, fourth one, parentenic host. Where the parasitic reaches the sexual maturity, that is called as the definitive host. Where asexual development occurs, that is known as the intermediate host. Maintains the parasitic in nature, it is called as a reservoir host. No development occurs but may aid in transmission, it is called as the parentenic host. Next, we know about the mode of transmission, how parasites transmitted from one person to other person and cause the infection, that is called as the transmission. First one, contaminated food and water, example parasites Endamoeba, Giardia. Next one, insect bites, that is example of the bites Plasmodium via mosquitoes. Skin penetration, cystoma is transmitted through the skin. Ingestion of undercooked meat, tinea. Example, tinea. Next one, congenital or transfusion transmitted and cause the infections. Last one, how we diagnose the parasites. Various methods used to detect the parasites in humans, both in the lab and using modern technology. There are many techniques available. First one, microscopy. Stool and blood disease samples were used the microscopic technique. Second one, serological test. It is used to detect the antigen antibody reactions and identify the parasites. The third one is the molecular method. PCR is used for the molecular methods. Last one is the imaging techniques. These are the techniques used for the diagnostic the parasites. Conclusions. Medically, parasitology safeguards the human health by identifying parasites, studying host relationship, life cycle, diagnosing infections, and guiding the treatment and enabling the prevention. Public awareness and ongoing monitoring help reduce the parasitic disease burden and enhance the community health. I hope this session is very useful for know about the what is parasites and how it is transmitted from one person to another person, and what are the methods we used in the diagnostic. Thank you for watching this session.